Hey, everybody. Welcome. Welcome to STAT's virtual event, STAT at JPM, Facing 2021, sponsored by Syneos. Thanks for joining us. I'm Matthew Herper, senior writer for medicine here at STAT. And in just a few moments, I'll be joined by our featured speakers to discuss what's next for the pharmaceutical industry after this difficult and unprecedented past year. But first, a few quick housekeeping items. At the bottom of your screen, you should see a few icons. Use the chat feature to chat amongst yourselves or to send a message directly to the STAT team who are using the handle STAT Events. Please do not put your questions in the chat. We do want your questions. Please do ask them and use the Q&A feature to type your question. We'll try to answer it live. Realize that there are a lot of you and there may be a lot of questions. We won't get to all of them. In the Q&A window, you should also be able to see the questions that other attendees have asked and upvote the questions you would most like to see answered. We will be paying attention to which questions have the most upvotes. We'll be taking questions throughout the chat, so please don't wait until the end to submit. This is part of what makes these events fun. Thanks to all of you for being here. For the first part of today's editorial program, we will be taking a look behind the first COVID-19 vaccine to be approved, produced by Pfizer and BioNTech. Here to discuss this breakthrough in medicine, please welcome Dr. Ugur Sahin, Chief Executive Officer and co-founder of BioNTech, and Dr. Özlem Türeci, Chief Medical Officer and co-founder of BioNTech. Thanks to both of you for joining me today. And we have both of you. Fantastic. I will note for the audience that in addition to being partners in business, you guys are partners in life and have been married for quite a while. You know, one of the things about this pandemic is I usually like numbers quite a bit, but for a lot of this year, numbers didn't feel like they were my friend. Every number that I saw about the pandemic made things seem worse. And that was true until we got to 95%, which was the vaccine efficacy of your vaccine and also really of the other mRNA vaccine made by Moderna. It's really one of the first bright lights we've had here. And I was just wondering for both of you, what was that moment when you heard that number like? Özlem, do you want to start?
I can start, yes, sure. Oh, it was a very emotional moment. You know, in the vaccine field, 95%, that's quite a number. I had not expected that. You know that normally, for example, with flu vaccines, we work with much lower efficacy. So it was a very emotional moment when we heard first and were informed that we have 95%, and there was a lot of jumping up and down and hugging each other and being very happy about that.
And Ugur, so what was that moment like for you?
Oh, it was an absolute relieving moment. We had, of course, all the immunogenicity data, and the immunogenicity of our vaccine was great. We knew we have T cells, we have antibodies, but there was a completely unknown part, whether the virus itself is amenable for control in humans. And so my expectation were in the range of maximum 80% to no activity at all. And getting this result was 95% efficacy rate. This was unbelievable and of course relieving, because we knew we have a solution for this pandemic, and this is just an incredible moment.
So why had you decided to embark on this? I mean, this was a big thing to feel the need to do, to make an mRNA vaccine. What was it that drew you to this problem, Ugur?
Yeah, so we are a cancer company, and we did not have infectious disease product in the clinical stage. We had the goal to develop infectious disease vaccines, but our main business is really cancer immunotherapy. We have personalized vaccines. And I read a paper which came out in Lancet describing this. It was the first really scientific paper describing this pandemic in a family who traveled to Wuhan, and there were a number of information pieces which were already known. But what was really annoying is the information that one of the family members had no symptoms, yeah, was asymptomatic but virus positive, and this virus fulfilled the pattern of a pandemic virus. It was a new virus known that to cause an epidemic 20 years ago in a family. It was spreading fast. It induced severe disease. Wuhan is one of the best connected cities in China, and it was for me when reading that very clear that this is not anymore local, and it will be extremely difficult to keep that, to control that. And the second idea was, hey, we have a technology and we have skills to extremely fast develop vaccines, and then it was very clear we have to embark into that.
Özlem, tell me about hearing that idea after Ugur had read that Lancet paper. I mean, it's a big leap.
Yes, it is quite a pivot. As Ugo said, this was in January 2020, and we had just had our IPO a couple of weeks basically ago, and that was part of preparing to accelerate our oncology pipeline. And I had used the holiday break to make plans. Because this year would be about accelerating the pipeline, bringing new immunotherapies we had developed into clinical stage and advancing those which were already into randomized trials, and extending our clinical trial footprint geographically and so on. So I had a lot of best laid plans how to use 2020 in an optimal way to bring our oncology trials forward, and therefore it seems like a crazy moment that at some point Ugur told me about the Lancet paper, and that he sees a threat at the horizon, and this threat would not only be a threat for our best laid plans for our oncology program, but it would also mean that this could be an opportunity to contribute to a solution. And it was a crazy moment, but at the same time, I know Ugo for many years, for 30 years now. So if he comes up with something crazy with this very earnest voice he had, and also explains the background why he thinks that we are close to a pandemic and we have to contribute. I don't hesitate. I follow, and it was quite a quick decision for me, but also for the entire company, to follow his lead and start this amazingly ambitious program from scratch to develop a vaccine.
Now, one of the things about the pandemic is that every time we have a triumph, there's something else. When we rarely have a triumph, there are other things to worry about. And the top questions here are about one of the things we were talking about before, which is the Stephanie Maris asks, what is your view of the one-dose strategy now adopted by HHS? Now, we were talking about this before, and there are a lot of different ideas. There's, do we hold all the doses? Do we, but the big question is do you need the second dose, and do you need the second dose on time? Ugur, I'd love to hear, I think the audience would love to hear your thoughts on this.
It is very clear that our vaccine is a two-dose vaccine, and there are several reasons why we need a second dose. The first reason is that we get a boost with the second dose for neutralizing antibody responses, but also for the T-cell response. And in the animal models, we clearly see that the antibody response is important to prevent infection. Yeah, you might be able to prevent disease with T cells alone, or to reduce disease with T cells alone, but strong neutralizing antibody titers are required to prevent disease, and this is very important. And we have only evidence for this protocol with prime boost 21 days apart. Of course, there is this discussion whether it could be a benefit for the society if the second dose is delayed and more people are vaccinated. And this is a risk-benefit analysis of the governments and health institutions. It will be, of course, that the time, if the time is delayed to the second dose, there is a risk, because we don't have data, that the protection which we see starts after 12 days could go down, yeah, if the second dose does not come after 3 weeks or 4 weeks or 6 weeks. Yeah, so there is a risk that we see a decline. I don't doubt that we will have a boost with the second dose, regardless whether this is 3 weeks, 4 weeks, or 6 weeks, because we know that from other protocols that this could be possible. But in the absence of evidence, yeah, we would of course favor, yeah, that the second dose is applied not much later than what is originally proposed.
Mm-hmm. There's a question here that I feel like I should throw in here, which is there a danger, this is from Pamela Katz, is there a danger that a one-dose strategy will result in resistant strains of the virus? Do we know?
This is an interesting question. We don't know. We have to be careful to speculate. We don't know. We have no. So we could even have resistant strains with a two-dose vaccine. So the two-dose vaccine does not mean that this is because of course if we go into a situation where sufficient people are vaccinated,
which is what we want.
Yeah?
Which is what we want, to be
what
clear.
We want. Yeah? Of course, this creates selection pressure. Yeah, and selection pressure means selection of variants which might, but this could come many years later, or 1 year later, or 2 years later. It will not happen directly. So I can't answer the question, to be honest.
Mm-hmm. Evolution's a powerful thing to be up against here.
Yeah.
So another issue to the other point, Özlem, has been that in multiple countries, the distribution here of the vaccine hasn't gone as well as one would have hoped. In the US, we've not only had difficulty getting it into arms, we've had some doses expire and have to be thrown out. How does that look from your standpoint, watching?
Yes, on the one hand, for a team as ours, which has developed the vaccine and definitely wants each and every dose to be used according to the purpose, it's disappointing to see when things don't go smooth in terms of distribution. Or vaccine doses go to waste. On the other hand, we also have to keep in mind that also the distribution part and the logistics, irrespective of which vaccine we are talking about, the distribution in the context of a pandemic is something which has never been done before and not practiced. There were no dry runs. It's also a gigantic endeavor and challenge, and we cannot expect that it goes smooth from the very beginning. There's also no one to blame for this. I'm sure and I believe that once all the stakeholders involved gain some experience, things will go smoother. And the most important thing is really to learn from what we are encountering in the process and act upon it.
Another big question that we're getting from the audience is asymptomatic spread. When will we know if the vaccines prevent asymptomatic spread, and to what degree? Can you still carry the virus and spread it, even though we know the vaccine does a great job at preventing symptomatic disease?
So we know from animal experiments that neutralizing titers, high neutralizing titers, are relevant and enable protection from infection. Yeah, and protection from infection is the key to prevent transmission. And because if there is an infection, the cells will produce new virus, and this is the basis for transmission of the virus to the next one. So we will get some data about asymptomatic infections in February, yeah, and we are excited to share that as soon as possible. Yeah.
Another thing that's been a big question has been the issue of allergic reactions to the vaccine, which obviously in a pandemic, people should still be getting the vaccine, but it has been a cause for concern. And it looks like there may be a rate as high as 1 in 100,000 people, which is pretty rare, but higher than, say, a flu vaccine. What are your thoughts on that? And I know you mentioned in your JP Morgan presentation, new formulations that, for instance, might not have PEG and might also be more shelf stable. So what's the status of bringing those online?
If I may first answer to the allergic reaction part, and Ugo, you might
that sounds sure.
Comment on the PEG. In the clinical trial, in the phase 3 trial with 44,000 participants, we have not observed anaphylaxia. In the rollouts now, in which in the meantime millions have been vaccinated, we have observed reactions. What we cannot say at the moment is what the incidence really is, because we and also health institutions like CDC and so on are looking into each of those reported cases and have to gauge whether these are indeed anaphylaxias. There is a process through which all of these cases have to go and have to be validated. And only if this process is basically through, we can comment on frequency or prevalence of anaphylaxias and answer the question whether they are higher than expected. This process is still ongoing. Mm-hmm. So you might want to say something with regard to the PEG?
Yes, so if this is the case, if we have really a higher rate of allergic reaction as compared, for example, for the classical flu vaccine, one hypothesis is that the PEG is the responsible part, and this is well known that PEG has antibody responses, but antibody responses are not explaining these reactions. We have the opportunity to leave out PEG. We have already described last year PEG-free formulations, LNP formulations, and we have a number of research programs dealing with next generation formulations, including those which do not have PEG. So that means that this could become a next generation formulation if needed, to come up with a formulation that does not carry a PEG element.
What are the steps for a next generation formulation? With some drugs, you can make formulation changes fairly easily. Other times they require more human studies. Do we know where we are with mRNA? It is such a new technology. You're the first ones to go through a regulatory process here, right?
Yeah.
So,
yeah. So the formulation research is ongoing, and we are working on formulations, for example, lyo formulations, or more stable liquid formulations, and the field will move fast. So there is no reason why an mRNA mRNA formulation should be different as compared to an antibody or a viral vaccine formulation. So it is because it is the first time that we are moving with a product so fast to the market. There was just no time to come up with something which is already fully mature. So the field will make big leaps in the next 12 to 18 months, coming up with formulations that are not different than those which are used for traditional vaccines, including lyo formulation, including formulations which might be even stored at room temperature or at 2 to 8 degrees for several months. Yeah.
Wow. Okay. Another question kind of along these lines from Esther Dyson, on asking what the impact of the interval between doses is for the 2-dose protocol, which you've already said you don't know, but she says based on other vaccines. And I do wonder how different, I mean, one thing that does seem very different about the mRNA vaccine versus some other vaccines is that you get so much of both your immune boost and even the reactions, the kind of fevers and stuff on the second dose. How predictive is the experience from other vaccines for an mRNA vaccine as we deal with these questions? Because it does seem different from, say, a protein subunit in some ways.
Yeah. I can start and you can add, Esther. So what is special for a messenger RNA? The messenger RNA, one special aspect is really that you can boost and boost and boost. So in our oncology trials, we have boosted patients up to 20, 25 times. Yeah. And every time when we boost the immune response. And the special mode of action of this boosting is that the protein is not visible for the immune system until the vaccine is delivered and the protein is produced by the cells. And that provides a wonderful mechanism for boosting. And this boosting is limited for viral vector vaccines because the vector is already visible to the immune system. And this is also a little bit limiting protein-based vaccines because the antigen, before it reaches the lymph nodes, is recognized by existing antibodies, and therefore this is, for example, one of the reasons why you need this relatively high amount of proteins for the second and third vaccination. I think this is a key advantage for messenger RNA vaccines from the mode of action, and this also predicts us that messenger RNA vaccines might become one of the key technologies for the booster approaches. We know that immune responses will decline over time, and mRNAs might be the best position to address the need to boost the immune response every 1 year or every 2 years.
Do you have something you wanted to add to that, Özlem, or should we?
No, I think Ugo has covered it.
So we have two questions here that I want to use to launch into a slightly different topic, which is, one is mRNA is a breakthrough for vaccine technology. What might the next breakthrough be? And another person asked, what application of mRNA vaccines excites you the most? And when we were speaking earlier, Ugo, you said that you don't think of BioNTech as a mRNA company. You think of it as, you think of yourselves as next generation immune engineers. So let's step back a bit and talk a bit about the other projects you're doing and the other types of potential that you see here, both for the cancer pipeline and also for other things like flu and autoimmune disease. Özlem, do you want to walk people through the cancer approach first? All that stuff you had to put on hold at the beginning of the year.
Yeah, we have not put it on hold, but it was obviously due to lockdowns and so on, and patients not be able to travel. It was slowed down quite a bit. So we have other platforms as well. Our main motivation is that we want to use immune mechanisms against cancer, and that means that mRNA for us is one way to use immune mechanisms in the context of vaccines, for example, but we also have other approaches. One of the approaches we are very excited about are our CAR T cells, meaning autologous cells of the patient who are engineered to express chimeric antibody receptor, which is directed against a cancer target, a very selective one. We have identified cancer targets which are highly selective for cancer cells, so that one can avoid any collateral damage, and will in a couple of weeks have a first novel CAR T therapy in clinical testing. So this is an approach we are very excited about. Ugo, do you also want to add one of your favorite platforms?
Yeah, so we are particularly excited that we have a plethora of combinatory approaches. We have, for targeting of cancer, we are using monoclonal antibodies, RNA-encoded antibodies, vaccines.
Okay, stop. Explain to people what an RNA-encoded antibody is. If we're going to geek out, let's go there. Explain an RNA-encoded antibody.
Yeah, so an messenger RNA encoded antibody is an antibody which is produced in our body after injecting the mRNA. And it is not just simply about instead making an antibody in the cell culture and purifying it and giving it to the patient. Instead of doing that, just providing an mRNA which does the same. It is more, it is really more. And the exciting aspect about mRNA-encoded antibodies is that you can, for example, use antibody molecules, bispecific antibodies, which are difficult to produce. So bispecific development, antibody development takes years. And one of the challenges is that if you have this small so-called single chain bispecifics, they have really short half-life. Yeah, in patients. So you have to find a way to stabilize that. You can do that either by PEG or you can just infuse and have pumps. But if you use a messenger RNA, then you can just use the half-life of the messenger RNA and inject the antibody and then have a 1 week or 2 week circulating antibody. So you can increase the half-life. So this is a way in circumventing the problems of the challenges of traditional antibody development. And on the other side, also combining that with the opportunity to deliver multiple molecules in parallel. Yeah. So this is a very exciting field in our, and we believe that this could allow us to bring novel type of molecules, engineered antibody molecules or engineered cytokines. So we have ribose cytokines. It's the same. So this is a cytokine which is encoded by an mRNA. Again, it follows the principle increasing half-life and also targeting to certain tumor tissues. So this is we can go back now into into immunological concepts that have been described 30 years ago and revisit them and come up with solutions which have not been technically solved so far. And this is an exciting way to go with mRNA encoded biomolecules.
Let's, both you and Albert Bourla at Pfizer mentioned the project that brought Pfizer and BioNTech together, which is another potential pandemic, which is flu, right? What are the advantages of an mRNA vaccine over the existing flu vaccines that we have? Why make a new flu vaccine? We've had flu shots forever.
So I start, Esther?
Yeah.
Yeah, I think there are two promises. One promise is really what happens is every time when this flu season starts, the WHO defines some strains, and then there are several months time until vaccines are developed. And we know that the strains that have been selected for vaccine development might not those which hit the population later on, several months later on, because there is change of the strains in between. And one promise of the mRNA approach is that you can just wait longer until there is more certainty which strain is the right one. And so this is really exciting that you can come up with more informed selection of strains for developing a vaccine. And the second potential advantage is that we have seen now in elderly people with our vaccine that we have more than 94% of efficacy. Yeah? And we know that elderly people, particularly elderly people, provide a challenge to get high efficacy rate for flu vaccines. And the promise here is, and this has of course to be tested, that we could come up with much higher or better effective flu vaccines if we could use the same mode of action. But of course, these are two promises that have to be evaluated.
So we have that seems like a good place to get back to a question I'm seeing both in the Q&A and that I've heard a lot from people, which is I've seen things online that say that the studies did not include people over 65. They absolutely both did. But how are the COVID vaccines performing in people over 65? Özlem?
So for our vaccine, we can say that they nearly perform identical as compared to younger age groups. 40% of our participants in the phase 3 clinical trial were older than 55 years, and we had also quite a large portion of older than 65 years participants, and the efficacies are around 94%, 94.7% to be precise. And I also should add that one of the reasons why we chose this vaccine candidate was seeing in our phase 1 trials on the immunological level that the immune responses with this B2 candidate, which we then moved into phase 3 studies in the older and the younger adults, were comparable. That was one of the criteria why this vaccine was chosen. Yes, we have data, and we can show that it is equally efficacious in the older ones.
Another question from Ravi Chivakula, which I feel like I keep hearing. If all the participants of the vaccine trials maintained social distancing, masking, and stayed in their basements, is the efficacy data truly representative of the vaccine's real efficacy? And maybe we can throw in here the concerns about, well, did people behave differently because they were unblinded based on side effects and concerns like that? But kind of how much can we really trust these efficacy results?
Yeah, so this is really a huge study with more than 44,000 participants, and it is a triple-blinded study. So the participants do not know, the physicians did not know, and we did not know who is receiving the vaccine. And this is the absolute gold standard in performing such trial. Of course, some participants might speculate that they have received the vaccine, but if they think that they have received the vaccine, then there would be more a tendency to be less cautious and be exposed more, and that would that if there is a trend, then there would be a trend to against the vaccine efficacy. Yeah, and we have seen that this double-blinded or triple-blinded study gave this results, and this is the best experiment you can do. There is no better experiment you can make, and if there is a better experiment to be proposed, then we need to discuss that. That's what the industry does since many years. Yeah.
So, and then a question I'm getting here that I also hear a lot, and when I asked a clinical trialist, I think she was in your trial. I think she was one of your primary investigators, but I asked, what should I ask them today? And she said, when are we getting data in kids, and how young are they willing to go?
There is an entire pediatric program which is planned and also discussed and proposed by the regulators, by EMA and by FDA. We will start a dedicated clinical trial in the younger population in different age strata, which will include 0 to 15 years in March, latest April, and will conduct an immune bridging study. This is not for efficacy, but to show the same immune responses which we have shown protect adults in the phase 3 trial. We have already had 16 to 17 years in our global study, and the vaccine is therefore authorized for this age group as well. And we also had a smaller subset of 1,000 actively immunized 12 to 15 year olds. They were not in the original emergency use approval because they came later at the end of the study, but we have already data from this pediatric strata as well. So there is a broader pediatric program.
So another number, which was a happy number when I heard it, but I have somewhat more mixed feelings about, is the one you announced at your presentation, which is 2 billion. Or 2 million rather, which is the, sorry for the misspeak there. But the number, well, no, it was 2 billion. I wrote it down.
Yes,
right.
2 billion.
The number of doses that you'll have, 2 million would be, we've given more than 2 million. How confident are you that you'll be able to hit that? And what are the constraints on being able to have more?
So first of all, this was an approach which took us about 6 weeks understanding how can we overcome constraints and
Mhm.
produce more than 1.3 billion.
Can you explain a little bit what the constraints are too?
The constraints are on different levels, and so this is the constraints on the raw materials. Yeah, do we have enough lipids? Do we have enough nucleotides? Enough cap? So this is one part of the constraint. The second is can we produce sufficient messenger RNA with the raw materials? And the third one is what's to scale up the drug product, the LNP manufacturing, which is one of the greatest challenges. And the last one, of course, is fill and finish capacity. And we are, of course, in a happy situation that Pfizer has multiple sites, and we have multiple sites. And we bought a few months ago, or a few weeks ago, a manufacturing site in Germany. This is the ex-Novartis site in Marburg, and we started to transfer the projects. So we asked the question, can we overcome the material constraints? Can we overcome the production constraints? Can we produce more? And can we fill and finish more? And we went through this, and we did not leave any stone unturned, and came up with a realistic scenario. But this realistic scenario has a number of ifs. And of course, the ifs are, for example, increasing or getting more manufacturing sites into our network, which will depend on approvals by, for example, the European Medicinal Agency or by the FDA, by ensuring that all materials which are now ordered have the right quality and come in a timely fashion, but we believe that this number is really realistic, yeah, and we can deliver 2 billion. And another important progress was that we came up with a reliable strategy to get 6 doses out of our vials, which increased the capacity by 20%, and this was approved by FDA, by EMA, by WHO. So that this is a combination approach. We are confident that we will be able to deliver this up to 2 billion doses, yeah, in 2021.
All right. Well, fantastic. Thank you for being here. Thanks for developing a vaccine. I usually don't say that, but I think everybody kind of agrees on that one. So we really appreciate both of your time. We're about at time now. Thanks to all of you for all your questions. We'll be moving on to our next segment now. So thanks again, guys. I think Britt's
Thank you for having us.
It was wonderful. And now I'm going to welcome my colleagues, Adam Feuerstein, Damien Garde, and Kate Sheridan. And we're going to talk about all the news that's come out of the JPM conference this week. Guys, you here? Hey, Adam.
Hey, Matt.
Got your radio host mic up.
Yeah, I have to say that it's a little bit humbling speaking after the two people who were like saving the world.
Right?
I mean,
and
like,
they're pretty humble too, which is striking for people used to biotech executives.
Yeah, it's a little bit like, wow, okay, maybe we should have been the opening act, but okay.
I'll remember that for next time. Well, there are a lot of people still watching, so how's your virtual JPM been, guys? How's this? How's doing this over Zoom? I know I kind of had the photo of me running from a cop on a Segway from last year come up in my Facebook memories, and did kind of miss San Francisco, even though I said I wouldn't.
Damien, when do you start?
I was going to say, I mean, it doesn't feel comparable whatsoever. I mean, going to JP Morgan has been an annual tradition tradition makes it sound maybe a little merrier than it is, but an annual workplace obligation for me for so many years. And I don't feel like I'm doing a virtualized version of that. I feel like I've just skipped it. The meetings I've done or presentations I've listened to have nothing in common with the JP Morgan experiences as I've come to know it, such that to say like, how's my 2021 JP Morgan? It feels like we've deferred or taken a rain check or something. Like I wouldn't even know how to compare them.
If I didn't have kids who were growing, I don't know that I'd feel like this year had happened at all. So,
Damien, someone in the comments thinks that you need a haircut.
Well, I think that's the poll we're running later in this.
We can talk about that
not
for the
just
rest of
someone
like
in
the
the
20 minutes we
comments,
have.
Mark McInerney.
Yeah. Hey, Mark.
Oh.
Mark.
Just to echo what Damien said, I think it is hard. It was always going to be difficult for a virtual event to sort of be the same as a live event. And I think that's been reflected this week. I know, and maybe Damien can talk to this because Damien and I have sort of been probably more clued in or tuned in to the presentations. Like, it's just really hard to be on a Zoom thing all day. I think we all had schedules. I had a lot of companies that I wanted to hear on Monday, and that's typically what it is because some of the bigger companies present on Monday. And so, you're there and you're listening, and it's, by the afternoon, you're just like, ugh, get me off this computer. And then, for the rest of the days, it is sort of like, so I think it's hard. And when you're live, at least you're in a room, there's like a physical, there's like a physicality to it. You're talking to people, you're running into people. So I think it's just hard. Like, it's not the same. I agree with you. It's just not, it's never going to replace, the live thing.
Can I say something? Because I think this year is an interesting counterpoint to last year's discussion around JP Morgan, because like everyone then was talking about like, oh, maybe I'll skip it. I don't need to be there. I can do meetings in Boston, or I can do things outside of the conference themselves. And so I'm curious because, for the, in the interest of disclosing this to our audience, this is my first JP Morgan, like formally speaking. I was not out in San Francisco and have not been to San Francisco before. And so I guess I'm kind of curious from your perspective, guys, do you think this year might have taught something to those folks who thought they could skip it last year, I guess, or thought that maybe this year they would do it virtually?
We actually like the parties. We just have to admit it. I think that's the general. That's why we have a super spreader event every year. I do want to get on to some of the topics we can get back to talking about JP Morgan, but I do want to get to some of the things that did happen this week, because there was actually some news. There was one case of real M&A that I remember, which was the Sanofi deal. But I think the biggest news that came out during JPM, and Dan Skowronski at Lilly swore to me that this was completely coincidental, that this was Monday at JPM, but it was. And that's, of course, the Lilly Alzheimer's news and the resulting increase in the stock price of also Biogen. So Damien, we're going to be covering beta amyloid for another 5 years.
Yeah, as we probably should have expected anyway. But yeah, Matt, to your point, the news is that Lilly ran a relatively small trial with an amyloid targeting antibody and saw a demonstrable and statistically significant benefit in terms of cognitive decline in patients with Alzheimer's, which, with the exception of you could substitute most of the nouns in that sentence and say it aloud in 2016, 2015, 2019, 2012, and obviously there are no approved amyloid drugs, which I guess is an important caveat to it. But yeah, I mean, I guess beyond my interest in the data, and we can talk about some of the implications, it did feel just kind of, I mean, like Joe Biden is going to the White House. There's economic concern, and it's just like 2009 headlines, but in 2021, I don't know.
McInerney just said seriously 2007. He's been a bear on the amyloid hypothesis for a long time. So
What about that endpoint that they used, guys?
It is so
Thoughts on that?
What they told me about that is that it lumps together two scales we're used to, which I think ADRS COG and one of the other ones, and simply adds them. But I really want to see the other. I mean, if it's as described, it's a much cleaner amyloid study than we've seen in the past. But we have literally 3 sentences in a press release. So I do really want to see the data there.
And Damien, you listened to the Biogen presentation. I did not. But it came up, right? I mean,
it sounds like it really came up.
I mean, like, was this like the best news ever for Biogen?
I mean, I would defer to Biogen itself, but yeah, it came up in the Q&A. I think Michelle Vanatzos, the CEO, made a brief mention of Lilly's data during their presentation, but I mean, it's sort of a heads I win, tails you lose kind of when you're in Alzheimer's. Whenever anything happens, if it's bad to a competitor, then it just illustrates why the differences, however infinitesimal, between your drug and their drug are so important. If it's good for a competitor, then it just illustrates the strong similarity, like, whatever. What was, I don't know. I mean, so I guess it's important. You mentioned a different endpoint. Lilly used a different endpoint that we're not necessarily used to, and they didn't break out the components of it that we are used to, such that we could make anything akin to an apples to apples comparison. Their drug is different biologically from Biogen's in terms of the form, or without getting too deep, without the type of amyloid that it targets in the brain.
It's another sentence you can say again and again, Damien. It clears more amyloid than the last one, and it really seems to. So
yeah, it does. I mean, the thing is, I think another thing we should keep in mind with respect to caveats of this, clearing amyloid from the brain is not really the hurdle that we're yet to see surmounted by these companies. I think even the FDA panel that was very skeptical of Biogen's drug aducanumab, they all seem to agree. There was one question that was like, does it clear amyloid for the brain, in so many words? And that was, I think, the only one that got a plurality or a majority of panelists to sign off on. So I guess, yeah, I mean, it's mechanistically promising, it's small numbers, et cetera, but it's difficult to metabolize the decades of reasons for skepticism and just kind of run forward positively with the Lilly news. Consider.
So other than moving on from Alzheimer's for a second, and not talking about Damien's hair, Mark McInerney wants to know asked us about M&A. There were no big M&A deals. Matt, you mentioned the little Sanofi one, which was like $1 billion. So that was nothing.
$1 billion for friends.
Yeah, it's nothing. So what does this team think about current valuations? Team?
Kate.
I don't know. I mean,
I think they're high.
I don't necessarily think that's a bad thing for biotech companies right now. And I'm coming at this, I think, a little bit from the startups perspective, where it's like all of a sudden they don't need to be bought. They can just go public, because it seems like anyone
do they
do that
even
now.
need to go public, Kate?
Well, we were having this conversation earlier, I think, Matt. At a certain point, it seems like VCs have just raised so much money for their own funds that they have to put it somewhere eventually, right? They have 10 years to do so. I think the SPAC sponsors probably have a slightly more pressing timeline. So to their point, I think they might have to go public eventually. But I don't know. Valuations do seem awful high. But whether or not that's a bad thing, I think very much depends on where you're sitting in this industry. I think for startups, high valuations in an open IPO market probably sounds pretty good.
I think I think it depends on the perspective that you have. If you're an investor buying stock in these companies and the stocks keep going up, then yeah, you're happy. I was struck, and I'll mention a specific example. I was struck by Beam Therapeutics. So Beam Therapeutics, relatively new company, developing drugs based on the base CRISPR, CRISPR base editing, right? So pretty cool, new technology. John Evans, their CEO, presented yesterday. Great presentation, talked a lot about the technology, why it's, why it might be a better type of CRISPR and things they're doing. Their first clinical candidate they're going to file an IND for their first clinical candidate in the second half of this year. So they're still not in the clinic. It's a $6 billion company right now. Now that's not John's fault, right? John isn't out like pumping the stock and telling people that, they're undervalued and it should be higher. It's just the fact that the market today has valued a preclinical CRISPR company at $6 billion. And, we've talked about where we've written about the kind of the phenomenon of Kathy Wood and ARK Invest and the ARK-G and ARK-K ETFs and them buying up all these CRISPR companies and then people sort of jumping on that bandwagon. And I think that's a big part of what's gone on. But that sort of puts John and Beam in a little bit of a bind, I think. Don't you think, Damien, that if you're at this point and you're a $6 billion valuation company, like, I mean, God forbid if you stumble, what happens? And it's hard to grow into a valuation like that, I think. So, I mean, it is a little bit difficult for them.
Yeah, it seems like you can kind of only disappoint strictly in terms of market reaction, but I can imagine in many ways it's a good problem to have, developing new things, specifically something that's never been done before clinically, like base editing is very expensive. So if I were at Beam, I would, the glass half empty is, wow, we can really only disappoint in the eyes of at least the stock price. The glass half full is, when it comes time to raise money, we can do it much more easily and much more non-dilutively, or whatever the adverb is there, because of the height of the stock price. I mean, I guess, we've seen this, people speak about the genomics bubble. There were sort of bubblettes, maybe
it's
circa
taken
2015.
20 years for me to hear the words genomics bubble again, and that can't be a great sign.
That's true, but the winning ideas survived. It's easy to say that in hindsight, right? There's like a survivorship bias, but like
sometimes the winning ideas haven't happened yet. The last time there was a genomics bubble, there was no Illumina, and there is only one large sequencing company now.
That's true, and it's possible we will look back on the current climate of having Editas Medicine, CRISPR, Beam Therapeutics, whatever, all the number, and they will have some sort of consolidation event and or survivor Darwinian event will take place, and there will be an Illumina of genome editing. I have no idea, but
I think part of the reason, just to return back to the valuation point, the original question, part of the reason I think I struggle to say, yeah, these valuations are inappropriately high is because of this preclinical issue, right? Like Beam, we have no way really of knowing whether Beam is going to succeed or fail until they start those trials, and they haven't even started yet, right? Like we can't even talk about enrollment speed. We can't talk about any of these things that might be able to give us some kind of hint about whether or not this number can be lived up to, I suppose. I think that's part of the reason I struggle with it. And I've been thinking about this a lot in the context of Sana, especially, which filed to go public today, because they just have so many programs, right? If one of these things succeeds, sure, maybe they can live up to this valuation. And that just seems like a huge unknown that we won't even have a reasonable answer to for a long, long time still. So
to be fair to Beam, and I know we shouldn't just talk completely about Beam because John Evans' ears are probably burning right now. But the Verve thing you mentioned, Kate, they did have data in monkeys that they presented or disclosed on Tuesday. Durable 6-month data from their cardiovascular one-time, and it uses, and I bring up Verve because they use the Beam base editor as the foundation for their
that's a good point. I'd forgotten about Verve actually, which I don't know.
But
how,
it's in monkeys.
in monkeys.
So let's make sure that we keep saying the word monkeys, and that's what it's in.
Monkeys, yes.
Ryan Flynn asks, do you think we'll continue to see more gene therapies snapped up by big pharma in 2021? What do you guys think?
Well, Bayer, there was that Bayer news, right?
Well, they're just hiring everybody to work with Jude.
Mm-hmm.
So,
yeah, I was going to say, could you argue that one of the lessons of gene therapy is that the technology has advanced to where, not that it's like plug and play, but if you have money and some like intellect, you could kind of replicate what some of the early movers have done. And then the other lesson is that the really important thing is to have the capital to set up your manufacturing such that you don't experience some of the things we've seen early movers experience. And so you could argue that like Bayer is doing this right by hanging back and let everybody else take the risk and learn the hard lessons, and then remembering that they're a multinational German firm that essentially prints money, and then getting in at that point. So like, do you need to acquire a gene therapy company if you're a major pharma firm? Is Pfizer might win the race in Duchenne muscular dystrophy, for example, and they're Pfizer. I don't know, I just made that up, but it's curious. I wonder what the smart people in the room are thinking with respect to the best way to enter gene therapy if you're a legacy pharma company. That's my answer to that question.
I think that's a good answer. Kate, you've spent a lot of time with the diagnostics companies, particularly the Thermos of the world, who've sold a lot of PCR tests, unfortunately, this year, even though it feels like we still don't have enough of them and can't conduct them fast enough. What are your big takeaways there?
I mean, I think one of the reasons I wanted to watch these testing companies present in the first place was just kind of see how they threaded this needle of having actually kind of great revenue news during a really not great year, right? Like that's a very awkward position to be in, to say, hey, we made all of this money from this horrible pandemic. Now, granted, a lot of them decided that they wanted to go about it by saying, look, we got our stuff together. We pumped out more tests than we thought we ever could. This is not a public service per se, but like we did this in the public interest. And I thought that was an interesting approach. I think it is interesting maybe that that people have sold PCR tests, but one thing that I kept picking up on is a lot of companies that wanted to emphasize their long-term kind of ability to continue this kind of revenue emphasized how many machines they'd sold. It's less about selling the tests for COVID and more ensuring that potential future tests will be run on your platforms and with your machines. And so I did notice that being brought up in a lot of different testing presentations, which I think is something that is, as much as we might diss PCR, I suppose, that might be the long-term ramification of having all these tests sold, I guess.
You'll never hear me diss PCR.
Oh, I know, right?
I'm scared of the ghost of Carrie, too scared of the ghost of Carrie Mullis. He'll come in on a surfboard and dose me with acid.
Well, this is part of the reason why I grin a little bit when you're like, they've sold all these PCR tests. It's like, that sounds like a good thing to me right now, to be honest.
Great question from Kelly Close. To what degree have speakers shown you how aware they are of the real world? To which she adds, global health crisis, racial unrest, the impeachment, the Capitol. Has the real world? How much has the real world intruded into your JP Morgans this year? The stuff Kate just talked about really is the real world. Those were companies that were operating in the pandemic.
I feel like those are the exception, though. And Adam and Damien, you guys tell me if your experience has been different, but I feel like that was the exception rather than the rule.
I heard a few CEOs sort of start their remarks kind of, sort of acknowledging what was going on, particularly last week with the Capitol takeover thing. Damien, did you hear anybody?
Not that specifically, no. And then as to those other examples, I mean, the pandemic is the really obvious one, but I think it's come up when it's been pertinent to investor conference topics. So for companies developing vaccines, it's very obvious. For other companies that had commercial issues in 2021 that they, reasonably or otherwise blamed on effects of the pandemic. And then with respect to reckonings over racism in the United States, no. I mean, not this JP Morgan. I feel like even then, when I have heard it in a biotech context outside of last summer, when it was top of mind for everyone, it's come from companies whose projects relate to it in some fashion. So I'm thinking of companies focused on, for example, sickle cell disease, which disproportionately affects Black people. Then they'll mention kind of the legacy of Tuskegee and the need for overcoming medical racism of the past, but it's very much pertinent to their business. And the same thing with when the vaccine trials were enrolling. I'm not saying people are being disingenuous about their concerns for equality in the United States, but I think they look at places like JP Morgan as one where that topic, if it's broached, it's when it's really critical to whatever the dollars and cents thing you're talking about is.
I will say that on Monday, Jamie Dimon, who's the CEO of JP Morgan, and Ken Frazier, who's the CEO of Merck, had a lunchtime sort of fireside chat. It was off the record, so I could not tell you what they said, which sucks. I mean, but I can't tell you. But I can tell you that they did bring up a lot of these, sort of social issues, particularly Ken Frazier, including what happened in the Capitol last week. And, he was very, These made some very forceful, sort of thought-provoking comments that I can't share with you because it was off the record. But maybe you can ask him privately, and he'll tell you what he said.
So one, we do have one. Carrie Watson, Adam, wants to know if this is different from covering JPM remotely in a normal year. I used to always cover it remotely, and I'd say yes. What would your answer there be?
Yeah, it's different. Yeah, because at least if you choose to cover it remotely, like I did 1 year before this, you have all the people there talking, and then you can kind of tap into that. But you can't do that here because no one's there. And that sort of leads to the question. So Robert Merold asks, does this spell the end of JPM, or do I make reservations for 2022? I would say make reservations for 2022, but make sure they're refundable. I don't know how you guys feel about that.
2023, I think we'll probably be back in San Francisco in some form.
I think we all
we might know that we really just want to go to the parties. Esther Dyson says that being in SF gives you the opportunity to see homeless people on the street, which is something we have noted at previous JP Morgans. There was a great question in here that I thought would make a good closer, which was from Christina Campbell, what therapeutic areas are you optimistic on for 2021 and beyond? Does everyone have one thing from JPM they're optimistic about?
I feel like every year, if not necessarily directly asked this question, if I were asked this question, my answer would be neuroscience. And yet every year there's like not really that much to show for it in terms of like clinical success. But I do think, just looking into 2021, there are a lot of large and seemingly well-designed trials in depression, for example, and also movement disorders that may fail or may not. But each seems to be informed by failures in the past in what hopefully are meaningful ways. And so I think, the best case scenario is that a bunch of drugs will work and it'll be a brand new era for neuropsychiatry and neuroscience in general, or they'll fail and hopefully, it's sort of just ruling out possible explanations for previous failures is in many ways the scientific process. And I think 2021 will be big for that as a therapeutic area.
I think there was, I didn't see the details, but I think that there was a report that came out, some data that came out, maybe it was yesterday, about falling cancer rates. Matt, did you see that?
I didn't.
That the cancer death rates are plunging. And, obviously that's a large part due to the drugs that are being developed. So that's that's always an area. There's always, we probably write more about cancer than anything else.
And that's not just because we like writing about cancer. It is when you look at the number of drugs in development, it's some outsize figure. It was more than half the last time I looked.
Yeah, if I had to pick an area, actually, I would agree with Damien on neuroscience, just because there seems like there's a lot of really young interest in a way. Like there's a lot of startups that seem really interested in working on neuroscience now, and I think that that's important, especially given that pharma and larger companies tend to avoid it these days. I think that's still true. That seems hopeful. So that's what I'll say.
So for me, the answer would be liquid biopsy. We've got some tidbits from Grail and from Guardant this year, but with Illumina buying Grail and Exact Sciences buying Thrive earlier detection, they're planning to roll these tests out in some form this year. I can't see how the pandemic doesn't disrupt that in some ways, but we'll finally be getting to see more data, it sounds like, and both for detecting disease early, which is what we usually talk about, but also for being able to monitor people who've had cancer to see if their cancer returns and catch it early. I do still think this is a really important technology to keep an eye on. And I think we're approaching the closing thoughts.
I would just say, I think we sort of all maybe bemoan the fact that there's not a lot of news and maybe that it was light on the news. But I think you do have to realize that, behind the scenes, there were thousands of meetings going on over this week, Zoom meetings, and those will result in deals. There will be partnerships, there will be acquisitions that will be announced this year that we will find out, happened because two companies got together at JPM 21, right? It's just, that's, I think that will happen. So, it's a little bit different this year, and, we missed so the physicality of it, but from that perspective, I think that you'll definitely see some things that come out of this that are, that we will be writing about.
I think we also always complain that the deals weren't as big as we expected. So you have to put that innate bias in there.
I don't know if I think the one closing thought, and somebody asked this earlier, is that after all the grousing about from longtime conference goers that this is my last year and next year I'm going to stay home, now that we've all been forced to stay home, I do think it's underlined some of the unvirtualizable benefits of something like this. So I guess, we all got what we wanted, and obviously no one wanted the pandemic, but we all got the reprieve from JP Morgan that many of us said we longed for. And so, we'll see if that galvanizes more interest in actually meeting up again when it's possible.
Hey, Kate, do you want to go next year?
That's the thing, is I actually think I kind of do want to see what it's like normally. I feel like this is definitely not what I have heard about. And now I'm really, really curious to see and experience what I always hear about from you guys. So.
Well, one of my takeaways from doing JP Morgan's at Stat has been, as with the rest of the year, how great everybody here is. So I'm just going to take this moment to publicly say how great you all are to have as colleagues, and to say that we're going to end exactly on time, which I all know that I love. Thank you again to everyone here. We really do appreciate you being here. I'm checking to see if I have closing remarks that I am supposed to say here, or if we're just supposed to log off. I want to thank Syneos, our sponsor, and tell everyone to keep an eye out for our next Stat virtual event at the end of this month. We'll be bringing together a panel of experts to discuss what can be done to prevent a potential black market for COVID-19 vaccines. Got another one coming up I think is really interesting on mental health during the pandemic after that. More information can be found on our website. Be sure to sign up and to join us. We also have weekly events for subscribers, our Stat Plus conversations. Until we see you again, stay safe and be well. Bye, guys.